to see so many people here. So what we already heard from the previous speaker is that if we wait long enough, 50% of our patients with uveal melanoma will die, and primarily from liver metastases. We have no idea why it goes to the liver, but it would be very nice if we could predict very early whether you are going to be having problems later on in life. And there's been many attempts to this. So this was the first attempt to do a prognostication of patients with uveal melanoma based on histopathology. This is the calendar system, which basically looked at spindle cells versus epithelial cells and a combination hereof. But as pathologists are like ophthalmologists, they don't really agree upon anything, so it's really difficult to have something good diagnosis system for epithelial cells. And this is just the Danish data to show that the histological diagnosis is not really good at detecting anything for prognosis. So in many cancers, we know the TNM system, and for the eye tumors, there is no lymphoid system for the intraocular tumors, so the nodule system doesn't really work, but for the T and for the M, it is really interesting, and the Kivile group showed, validated later by other groups, that you can actually use the size and the anatomical position of the tumor to predict outcomes later on in life. And this is the real-time data, and the problem is the largest tumors, that's not really differentiated. There's no stratification between the T3B and T3C. So it might have something to do with the classification system, where you have the largest basal diameter and the height, and in some cases you actually see that there's a problem with, you have a higher tumor than the basal diameter, so if you correct for that, it looks a little better, and especially if you look at volume instead of just two measurements, and this is just a supposed how you could look at the tumor volume, and if we do that, we have this distribution of all the tumors, and if we kind of divide it into different standard diameters of the median, we have 5 different groups, and when we look at the survival compared to volume, they have very good stratification based on small tumors survive very good, big tumors survive very bad. But the real breakthrough came in this paper from Berlin with Bornfeld and Foster that showed that the real survival of melanoma is whether you lose one of your chromosome 3 or not. And they have a very high separation, so basically the patients with normal chromosome 3 survives, and the ones that have poorer loss of one survives not so good. And this is just to show that we also look at chromosome 8q, and when we do this, we can actually divide into 3 groups, one that has absolutely normal chromosomes, one that has both 3 and 8q gain, and this has either the loss of monosome 3 or gain of 8q. And when we combine that with the TNM system, we actually see that if you have normal chromosomes, despite of the size of the tumor, you don't really die. Whereas if you have a big tumor and you have chromosomal defects, you definitely have a very poor prognosis. But still, you can have a small tumor and have changes, and you will do actually okay. And the good thing is here that you can actually stratify. So if you only had the TNM classification for T2 tumors, you will have this survival curve, but now with adding on of the genetics, you actually see that you can really stratify into 2 groups. And this is just to show that it is truly independent risk factors. But there's many other factors. This is the TGA classification and also gene expression profiling. This is more used in the United States with the Castle Biosciences. But when you talk about genetics, it's important to differentiate between somatic mutations and germline mutations. So germline mutation is something that you have in every cell in your body, whereas somatic mutations is only found in the tumor. So if you want to say something about the tumor, you need to sample the tumor. So you need to get the genetic information from the tumor and not just from the blood. So prognostication is possible. You can use the TNM classification, you can use genetic, or you can use a combination of those. it is useful for the patients, not for the patients with the bad chromosome, but with the normal chromosomes. And we actually take consequence of that in Denmark. So if you have a normal chromosome test from your tumor, you are not being followed in the same way as if you have an abnormal chromosome test. And it can be useful for clinicians in the future to distinguish between where should I use my resources. Should I use my resources on all the patients or just a subgroup which actually have need for it? Thank you.
I think we have time for a question. There's a question from the audience. Thank you, Professor.
All very clear and perfectly in time. So we go back to treatment and the treatment of choice